In this video, we are going to learn about the different pathways associated with apoptosis, like the extrinsic pathway and the intrinsic pathway. So first, let's learn about the extrinsic pathway. The stimulus for apoptosis is carried by an extracellular messenger protein called tumor necrosis factor, which was named for its ability to kill tumor cells. TNF is a trimeric protein produced by certain cells of immune system in response to adverse conditions such as exposure to ionizing radiation, elevated temperature, viral infection, or toxic chemical agents such as those used in cancer, chemotherapy. The stimulus for apoptosis is carried by an extracellular messenger protein called a tumor necrosis factor, which was named for its ability to kill tumor cells. TNF is a trimeric protein produced by certain cells of the immune system in response to adverse conditions such as exposure to ionizing radiations, elevated temperature, viral infections, or toxic chemical agents such as those used in cancer, chemotherapy. Like other types of first messengers, TNF evokes its response by binding to a transmembrane receptor, which is termed as TNFR1. The trimeric TNFR1 protein is a member of a family of related death receptors that turns on the apoptotic process. The cytoplasmic domain of each TNF receptor subunit contains a segment of about 70 amino acids called a death domain. Each segment, death domain, binding of TNF to the trimeric receptor produces a change in the conformation of the receptor's death domain, which leads to the recruitment of a number of proteins. So, there are death receptors that turns on the apoptotic process. The cytoplasmic domain of each TNF receptor subunit contains a segment of about 70 amino acids called a death domain. Binding of TNF to the trimeric receptor produces a change in conformation of the receptor's death domain, which leads to the recruitment of a number of proteins. The last proteins to join the complex that assembles at the inner surface of plasma membrane are two procaspase 8 molecules. These proteins are called procaspases because each of this is a precursor of the caspase. It contains an extra portion that must be removed by proteolytic processing to activate the enzyme. The synthesis of caspase as proenzymes protects the cell from accidental proteolytic damage. Unlike most proenzymes, procaspases exhibit a low level of proteolytic activity. According to one model, when two or more procaspases are held in close association with one another, they are capable of cleaving one another's polypeptide chain and covering the other one molecule with a fully active caspase. The final mature enzyme, that is caspase 8, contains 4 polypeptide chains derived from 2 procaspase precursors, and activation of caspase 8 is similar in principle to the activation of effectors by a hormone or growth factor. In all of these signaling pathways, the binding of an extracellular ligand causes a change in conformation of the receptor that leads to the binding and activation of proteins situated downstream in the pathway. Caspase 8 is described as an initiator caspase because it initiates apoptosis by cleaving and activating downstream or executioner caspase that carry out the controlled self-destruction of the cell. So when we just detailedly explain the process of extrinsic pathway of apoptosis, the stimulus for apoptosis is carried by an extracellular messenger protein called tumor necrosis factor, which we just shortly represent as TNF, which was named for its ability to kill tumor cells. Tumor necrosis factor. This is a trimeric protein produced by certain cells of the immune system in response to adverse conditions such as exposure to ionizing radiations, elevated temperature, viral infection, or toxic chemical agents such as those used in chemotherapy. And this TNF is produced by immune cells of our body. due to some conditions which are that much difficult for the cells, like high temperature, ionizing radiation, toxic chemicals, etc. When all these conditions are present, immune system produce certain cells which produce proteins called TNFs, which acts as a stimulus for apoptosis. Like other types of first messenger discussed earlier, TNF evokes its response by binding to a transmembrane receptor, TNF-R1. The trimeric TNF-R1 protein is a member of a family of related death receptors that turns on the apoptotic process. The cytoplasmic domain of each TNF receptor subunit contains a segment of about 70 amino acids called a death domain that mediates protein-protein interactions. Binding of TNF to the trimeric receptor produces a change in conformation of the receptor's death domain, which leads to the recruitment of a number of proteins. The last proteins to join the complex that assembles at the inner surface of the plasma membrane are two procaspase 8 molecules. These proteins are called procaspases because each is a precursor of a caspase. It contains an extra portion that must be removed by proteolytic processing to activate the enzyme to turn that into a caspase. The synthesis of caspases proenzymes protects the cell from accidental proteolytic damage. Unlike most proenzymes, procaspases exhibit a low level of proteolytic activity. According to one model, when two or more procaspases are held in close association with one another, as they are capable of cleaving one another's polypeptide chain and converting the another molecule to the fully active caspase. The final matured enzyme, that is the caspase 8, contains 4 polypeptide chains derived from 2 procaspase precursors. Activation of caspase 8 is similar in the principle to the activation of the effectors by a hormone or growth factor. In all of these signaling pathways, the binding of an extracellular ligand causes a change in conformation of the receptor that leads to the binding and activation of proteins situated downstream in the pathway. Caspase 8 is described as an initiator caspase because it initiates apoptosis by cleaving and activating downstream, or executioner caspase that carry out the controlled self-destruction of the cells as described. Now, let's learn about the next pathway, that is the intrinsic pathway of apoptosis. Internal stimuli such as irreparable genetic damage, lack of oxygen like hypoxia, extremely high concentrations of cytosolic Ca2+, viral infection, ER stress, or severe oxidative stress in the case of a production of a large number of destructive free radicals trigger apoptosis by the intrinsic pathway. Activation of the intrinsic pathway is regulated by members of the BCL2 family of the proteins, which are characterized by the presence of one or more small BH domains. BCL2 family members can be subdivided into 3 groups, which promote apoptosis, like BACs and BAC, anti-apoptotic members containing several BH domains that protect cells from apoptosis, like BCLXL, BCLW, and BCL2. And 3, BH3 only proteins, so called because they contain only one BH domain, which produce and promote apoptosis by an indirect mechanism. According to the prevailing view, BH3 only proteins like BID, BAD, PUMA, and BIM can exert their pre-apoptotic effect in 2 different ways depending on the particular proteins involved. In some cases, they promote apoptosis by inhibiting apoptotic BCL2 members, whereas in other cases, they promote apoptosis by activating pro-apoptotic BACs or BAC. In either case, the BH3 only proteins are the likely determinants as to whether a cell follows a pathway or survival or death. In a healthy cell, the BH3 only proteins are either absent or strongly inhibited, and the anti-apoptotic BCL2 proteins are able to restrain pro-apoptotic members. The mechanism by which is debated. It is only in the face of certain types of stress that BH3 only proteins are expressed as activators, thereby shifting the balance in the direction of apoptosis. In the circumstances, the restraining effect of the anti-apoptotic BCL2 proteins are overridden, and the pro-apoptotic protein BAX is free to translocate from the cytosol to the outer mitochondrial membrane. Although this mechanism is not entirely clear, it is thought that BAX molecules or BAC molecules, BAK, which are permanent residents of the mitochondrial membrane, undergo a change in conformation that causes them to assemble into a multi-subunit protein-lined channel within the OMM. Once formed, this channel dramatically increases the permeability of the mitochondrial membrane and promotes the release of certain mitochondrial proteins, most notably cytochrome C, which resides in the intermembrane space. Nearly all of the cytochrome C molecules present in all of a cell's mitochondria can be released from an apoptotic cell in a period as short as in 5 minutes. Cells lacking both BAX and BAC are protected from apoptosis, revealing the essential role in this releasing the essential roles of this pro-apoptotic proteins in this process. Release of pro-apoptotic mitochondrial proteins such as cytochrome C is apparently the point of no return. That is an event that is irreversibly commits a cell to apoptosis. Once in the cytosol, cytochrome C forms part of a wheel-shaped multi-protein complex called the apoptosome that also includes several molecules of procaspase 9. Procaspase 9 molecules are thought to become activated by simply joining the multi-protein complex and do not require proteolytic cleavage. Like caspase 8, which is activated by the receptor-mediated pathway described above, caspase 9 is an initiator caspase that activates downstream recruitment caspases, which bring about apoptosis. The extrinsic or receptor-mediated and the intrinsic, that is the mitochondria-mediated pathways, ultimately converge by acting the same precursor caspases which cleave the same cellular targets. You might be wondering why cytochrome C, a component of the electron transport chain, and the mitochondrion, an organelle that functions as a cell's power plant, would be involved in initiating apoptosis. There is no obvious answer to this question at the present moment. The key role of mitochondria in apoptosis is even more perplexing when we consider that these organelles have evolved from prokaryotic endosymbionts and thus that prokaryotes do not undergo apoptosis. As cells execute the apoptotic program, they lose contact with their neighbors and start to shrink. Finally, the cell disintegrates into a condensed membrane-enclosed apoptotic body. This entire apoptotic program can be executed in less than an hour. The apoptotic bodies are organized by the presence of phosphatidylserine on their surface. Phosphatidylserine is a phospholipid that is normally present only on the inner leaflet of the plasma membrane. During apoptosis, a phospholipid SCAM-base moves phosphatidylserine molecule to the outer leaflet of a plasma membrane where they are recognized as an "eat me" signal by specialized macrophages. Apoptotic cell death occurs without spilling cellular content into the extracellular environment. This is important because the release of cellular debris can trigger inflammation, which can cause a significant amount of tissue damage. That is something happening in case of necrosis and signaling cell survival. Just as there are signals that commit a cell to self-destruction, there are opposing signals that maintain cell survival. In fact, interaction of TNF with the TNF receptor often transmits two distinct and opposite signals into the cell interior, one stimulating apoptosis and another stimulating cell survival. As a result, most cells that possess TNF receptors do not undergo apoptosis when treated with TNF. This is a disappointing finding because it was originally hoped that TNF could be used as an agent to kill tumor cells. Cell survival is typically mediated through the activation of a key transcription factor called NFKB, which activates the expression of genes encoding cell survival proteins. It would appear that the fate of a cell, whether survival or death, depends on a delicate balance between pro-apoptotic and anti-apoptotic signals. Apoptotic cell condensed fragmented chromatin is there. The electron micrograph of a Clearance of an apoptotic cell is accomplished by phagocytosis. Apoptotic cell corrupts within the cytoplasm of a phagocyte. And we just look at the intrinsic mitochondria mediated pathway of apoptosis. We can learn that internal cellular damage is happening, activation of pro-apoptotic BH3 only protein and subsequent activation of BAC or BAX. Then BAX is activated, cytochrome C is released, and cytoplasmic factors and procaspase 9 is there. Formation of apoptosome like activated initiator caspase 9 complex. Execution of procaspase will be there, and then execution of caspase, and then apoptosis will be happening in case of internal circular damage like the intrinsic pathway of apoptosis. Various types of cellular stress cause pro-apoptotic members of the BCL2 family of proteins, either BAX or BAC, to oligomerize within the outer mitochondrial membrane, forming channels that facilitate the release of cytochrome C molecules from the intermembrane space. Once in the cytosol, the cytochrome C members form a multi-subunit complex with a cytosolic protein called APAF1 and procaspase 9 molecules. Procaspase 9 molecules are apparently activated to their full proteolytic capacity as a result of a conformational change produced by association with APAF1. Caspase 9 molecules cleave and activate execution of caspase, which carry out the appropriate response. The intrinsic pathway can be triggered in some cells, hepatocytes, by extracellular signals. This occurs as the initiator caspase of the extrinsic pathway, caspase 8, cleaves a BH3 only protein called bit, generating a protein fragment T-bit that binds to BAX, inducing insertion of BAX into an OMM and release of cytochrome C from mitochondria. OMM is nothing else but the outer mitochondrial membrane.